recent. Oh, okay. So this is the largest Art Nouveau precinct in the world, and our laboratory up until a couple of years ago was based in this precinct here. But 2 years ago, we built a new laboratory, which is actually a bit more modern and updated. It helps with the research, but it's just it's just out of frame on the first picture. So we're still at the Hospital São Paulo. So if anyone wants to come visit, it is a beautiful place to visit. So the head of the memory unit is Alberto Yeo, and our research group, as I said, is multidisciplinary. It has basic researchers, clinicians, neuropsychologists, geneticists, and there's over 40 of us. We are all interested in biochemical and neuroimaging biomarkers, molecular genetics, and the molecular basis of neurodegenerative diseases. And as I mentioned, my role in the group is to lead the synaptic biomarker research line. So in the first part of my talk, I'm just going to give you an introduction to the synaptic biomarkers, but the bulk of the talk will be on our latest data, including 3 clinical studies and some conclusions on where I think the field is going. So there's general consensus in the literature that neurodegenerative diseases are synaptic failures. Okay, so this was first proposed by Dennis Selko in 2002, in which he titled this paper, "Alzheimer's Disease is a Synaptic Failure." And this has since been recapitulated in many other neurodegenerative diseases. I'm just showing some examples here, but Parkinson's disease is believed to be a connectome dysfunction. There is a lot of evidence of synapse loss across the dementias, and also in other neurodegenerative diseases such as amyotrophic lateral sclerosis, Huntington's disease, and multiple sclerosis. And I'd also like to point out that synaptopathy itself is key to neuropsychiatric disorders such as schizophrenia and autism. So a biomarker of synapse degeneration could also be useful in neuropsychiatric disorders as well as neurodegenerative diseases. But my focus is on, for now, on the neurodegenerative diseases. And so let's start with Alzheimer's disease, the most common form of cause of dementia. And the most accepted hypothesis for Alzheimer's disease is the amyloid cascade hypothesis, which basically posits that the deposition of a beta peptide in the brain, what is called brain amyloidosis, initiates a chain of events which begins with synaptic failure, and also results in tau-mediated neurodegeneration, which leads to widespread loss and death of neurons. And these are in cognitive regions of the brain, and so this results in the dementia that is so characteristic of this disease. Now, Lewy body dementia is the second most common cause of dementia, and a similar cascade of events has been proposed. So Mahul Nair last year published in PNAS this paper in which they suggest that alpha-synuclein is endocytosed into the neuron, and it is then post-translationally modified, which initiates a cascade of events, which results in mitochondrial and synaptic dysfunction, both of which precede cell death. Now, Bellucci et al. In 2016 further went into the synaptic degeneration. They showed that synaptic damage and synaptic plasticity deficits are responsible for a connectome dysfunction, and it is this which causes the motor and non-motor symptoms that are characteristic of Lewy body dementia. And finally, frontotemporal dementia, which is the most common cause of early onset dementia. There is evidence that there is synaptic loss, particularly in the temporal cortex of FTLD patients. And also the C9orf72 gene, which is a genetic cause of frontotemporal dementia, is associated with decreased dendritic branching in primary mouse and rat neurons. And the implication is that this results in a decreased synaptic activity. So I think I can give you lots of evidence that synaptic loss is an early and core event in all of these neurodegenerative dementias. So it'd be great if we could measure this in living patients. Now, the cerebrospinal fluid offers a natural window into the brain. So it's an ideal fluid to measure changes that are going on in the brain, okay? And in terms of Alzheimer's disease, CSF markers are actually already used in the clinical routine. Okay, they're what we call the ATN biomarkers, A for amyloidosis, T for tau pathology, and N for neurodegeneration. And these are excellent diagnostic and prognostic markers for Alzheimer's disease. And we do not expect new biomarkers to improve upon these because they are measuring the underlying pathology. However, you'll notice from this slide, which is taken from Kai Bleno, who was published just last month in Science, there is a lack of biomarker for synaptic degeneration, which is actually the source of the clinical outcome. So in 2014, I studied a biomarker research line, the aim of identifying potential candidate synaptic biomarkers in the cerebrospinal fluid. So I can't go into details of this part of the paper, it's already published, but for questions of time, I'm just going to give you a brief rundown of how we did this. First of all, we defined the cerebrospinal fluid proteome by performing shotgun proteomics on pools of CSF samples from patients and controls. And we cross-referenced the proteins we identified with what we characterize as the synaptic proteome. We define that as proteins that have a known synaptic function and also are expressed at the synapse. And doing this, we identified 251 synaptic proteins that are detectable in human CSF. We then combined two techniques, which is array tomography microscopy and Western blot of synaptosome fractions, to show that a select set of these 251 proteins, we selected 9 for further follow-up, we showed that they were specifically expressed in the human cortical synapse. This, we took those 9 proteins forward and we developed a targeted mass spectrometry protocol, which allowed us to quantify these 9 proteins in parallel in the same sample, okay, which allowed us to evaluate their CSF profile in exploratory and validation cohorts. So the synaptic panel includes 9 synaptic proteins, which have a variety of functions as shown here. So ranging from synaptic vesicle-like exocytosis, dendritic spine assembly, calcium signaling, synaptogenesis, and presynaptic differentiation. So we first wanted to look in Alzheimer's disease, and we used samples from our own cohort here. And we picked cognitively normal controls who are negative for the ATN-AD biomarkers. And then we selected patients or subjects from across the preclinical and clinical AD continuum. So in the preclinical stage, they are cognitively normal, but positive for at least one of the AD biomarkers. And then we have mild cognitive impairment, which is called prodromal AD, and AD dementia. So we have a full continuum of preclinical and clinical AD. And we selected patients from across this continuum from 3 separate cohorts. And here we have the results. This is the mass spectrometry data expressed as log2 fold change at each disease stage compared to controls. Each color is a different protein, and the solid line is an adjusted p-value less than 0.05, and the dotted line is an adjusted p-value greater than 0.05. And this is the cross-cohort meta-analysis from the 3 cohorts. Okay, so for brevity in this talk, I'm just showing you the final cross-cohort meta-analysis. And what it nicely shows is that there is a clear pattern of all of the synaptic proteins in our panel, give or take, where in stage 1, okay, we see a reduction in the synaptic proteins compared to controls. Preclinical stage 1 is defined as shown at the top as cognitively normal, but positive for amyloidosis, which means they're already on the path to Alzheimer's disease, but they've yet to experience widespread neurodegeneration. And this is critical because, as mentioned earlier, this is a perfect window in which to try your therapeutic intervention. It's more likely to be effective before widespread neurodegeneration takes hold. Now, at later disease stages, what we see is actually an elevation in these synaptic proteins. And we don't know exactly what is going on here, but the hypothesis is that the low levels in the absence of neurodegeneration markers is reflecting reduced synapse density in the brain. But elevated levels over the course of the Alzheimer's disease continuum are actually reflect increased clearance of the synaptic proteins into the CSF. We were really interested in this preclinical stage, so we wanted to look at it in larger preclinical Alzheimer's disease cohort. So for this, we used our Down syndrome cohort. And you may wonder why Down syndrome? Well, most adults with Down syndrome develop Alzheimer's disease. If you see here from our paper we published in 2018, this shows the relative prevalence of Alzheimer's disease with age in adults with Down syndrome. And what you can clearly see is that from the age of 40 onwards, the prevalence of AD is gradually increases, such that by the time they reach the 7th decade of life, 90 to 100% of adults with Down syndrome have Alzheimer's disease. And what this opens up is a window of preclinical stage, as I mentioned earlier, before the clinical symptoms occur, but we know they're already on the route to Alzheimer's disease. So we have a prospective cohort of Down syndrome here in Barcelona. It's called the Down Alzheimer Barcelona Neuroimaging cohort. It's run by Juan Fortea in our group. And we selected cognitively normal non-trisomic controls as a comparison, and then Down syndrome subjects from across the AD preclinical and clinical continuum. And we quantified our synaptic biomarkers at all stages. Specifically, we wanted to correlate the synaptic markers to cognitive performance, okay? So we used 3 measures of cognitive performance. First of all, the CAMCOG score, and second of all, the cued recall test, which is modified for adults with Down syndrome. And in the cued recall test, it tests your immediate recall and your delayed recall. So we correlated our synaptic panel to these 3 measures. And as you can see here, the R-squared for correlation of these proteins. And it's clear that VAMP2 from our panel was the best synaptic correlate of cognitive performance in these individuals. And you can see the plots down below. Now, we wanted to make sure that this was actually cognitive decline and not intellectual impairment that we were measuring here. So we compared VAMP2 across in a Down syndrome cohort, dividing them by the severity of intellectual disability from mild, moderate, and severe and profound. And we found no association of CSF VAMP2 levels in each group. So we conclude that VAMP2, the changes in CSF VAMP2 are associated with cognitive impairment and not intellectual disability. We then wanted to look at whether these are related to Alzheimer's disease. So we compared CSF VAMP2 across the disease stages, as shown in panel A. And what you can see is similar to sporadic AD, the VAMP2 is reduced in the preclinical stage, shown here as ADS or asymptomatic AD, but relatively elevated in the symptomatic stages. This is also nicely shown in panel B, where you see the correlation with age in red in the Down syndrome cohort, where VAMP2 is subtly elevated in the older individuals. And finally, we showed that CSF VAMP2 correlated well with AD biomarkers. So we conclude that CSF VAMP2 is a surrogate marker of cognitive decline in adults with Down syndrome, and that this cognitive decline is related to Alzheimer's disease. So VAMP2 has also been implicated in synaptic degeneration in dementia with Lewy bodies. Okay, so we wanted to look at this in our cohort. We have the other cohort in our group, which is the SAMPOW Initiative in Neurodegeneration cohort. And in that, we picked 68 cognitively normal controls. We picked patients that have a clinical diagnosis of DLB or a clinical diagnosis of Alzheimer's disease. The DLB included both the prodromal and the dementia stage. And also we divided into those who had pure DLB and those who were positive for AD biomarkers. Now, this is a common occurrence in DLB patients where they show AD copathology, and so they're positive for the AD biomarker just as in the AD group. So we defined these the 4 groups, and we quantified VAMP2. And here you can see on the left that when adjusting for age, the VAMP2 was actually decreased in the pure DLB group compared to controls. However, it was elevated in the DLB with AD copathology group to similar levels as that we found in the AD group. Moreover, when we correlated CSF VAMP2 with a measure of cognitive performance, in this case, the clinical dementia rating, we found that only in the pure DLB group did we see a correlation such that individuals with low CSF VAMP2 and pure DLB were higher on the dementia rating scores. They were more demented than those with higher levels. We did not find the same association in those with AD copathology. So clearly, this shows that there are different mechanisms at play in terms of synapse degeneration in the pure DLB and the DLB patients with AD copathology. So in the final study, we wanted to look at the synaptic biomarkers in frontotemporal dementia. Okay, so for this, we used a neuropathological cohort. As Anna-Kasia mentioned, clinically, FTD is a very heterogeneous disorder with many syndromes, and it's very difficult to discriminate between them. And neuropathologically, there are many different disease subtypes, and they don't always correlate with a particular syndrome. Now, the Penn FTD and AD Research Center have a unique cohort which has ante-mortem CSF and post-mortem examination of these patients. So we were able to select 24 patients classified as having FTLD with tau pathology, 25 with FTLD TDP pathology, and 25 with AD pathology. And we also selected some cognitively normal controls, and we quantified the synaptic panel in each group. And what we find was interesting. There is 2 synaptic proteins, calcitonin 1 and neurexin 2A, which, when compared across groups, showed a reduction in FDLD compared to AD and controls that seemed to be specific to the TDP group. Okay, so this implies that low calcitonin 1 and low neurexin 2A levels in your CSF is indicative of having FDL-TDP. We saw a significant decrease in calcitonin 1 in the tau group, but only compared to AD, not compared to controls. So these findings indicate that maybe the levels of these proteins are related to TDP pathology. So we did a correlation analysis of the two proteins with the post-mortem TDP-43 burden. So this is ante-mortem CSF, correlating with post-mortem pathology, and specifically TDP-43 burden. And what we found was an inverse correlation. So this means low CSF before death, low CSF levels of these two proteins before death correlated with post-mortem burden of TDP-43. And we did not find the same in the with tau pathology. So this really suggests that low CSF calcitonin 1 and neurexin 2A are a surrogate measure of TDP pathology in the brains of patients. And this is really important because we are unable to measure the underlying pathology in these patients in life. So I think this is a really important finding. So to summarize our findings, CSF VAMP2 likely reflects different processes in Alzheimer's disease and dementia with Lewy bodies. In both cases, we were able to use it as a surrogate measure of cognitive decline, albeit in different directions. And in the case of Alzheimer's disease, where we were able to look in preclinical stages, we were able to detect very early pathophysiological changes. And this is very critical, as we've mentioned several times already today. The earlier you can detect these changes, the more likely you are to be able to set these patients for clinical trials and have a positive reaction to therapeutic intervention. CSF calcitonin 1 and neurexin 2A had a slightly different profile to VAMP2. While they showed the same nonlinear profile course over the AD continuum, they were specifically reduced in the TDP-43 group of FDLD, and so therefore could be used as surrogate measures of TDP-43-mediated synapse degeneration in FDLD. So the take-home message from these data, I think, is the following. Basically, synaptic biomarkers have a diverse range of uses in the clinical and trial setting. Specifically, they could be used as surrogate measures of cognitive decline or disease progression. And they can be used to detect early pathophysiological changes, specifically in certain subtypes. It's clear that synaptic biomarker profiles are complex and difficult to interpret, and they may not be linear across disease stages, but instead they may reflect different forms or stages of synapse degeneration across diseases. For example, we could be seeing the differences could be explained by synapse loss versus synapse dysfunction, possibly even compensatory mechanisms coming into play, or they could just simply be reflecting different regional differences in the expression of the synapses that we are quantifying. And I think really a very important aspect of these synaptic biomarkers and where I see the field moving forward, as I mentioned, in the case of Alzheimer's disease, we have excellent diagnostic biomarkers. We are not looking for diagnostic biomarkers, rather biomarkers that can help us understand better what is going on very early in the disease. And I think this has particular impact on clinical trial design. Okay, so there are no, well, in frontotemporal dementia and DLB, there are no drugs that are approved that can slow or stop disease progression. You may be aware that in Alzheimer's disease recently there was one approved. There is some controversy on this. But even in this, I've taken this figure from a paper published last month in which it was specifically referring to the clinical trials that were resulted in aducanumab, which is this drug that has been recently approved for Alzheimer's disease. And it was criticizing somewhat it was highlighting the limitations of clinical trials in these diseases. And the reason for this is, if we think of how these trials are designed, they have a disease-modifying treatment. In most cases, if you look here, most of these cases in these diseases, they are targeting the synaptotoxic agent, which they believe to be A-beta, tau, or alpha-synuclein, for example. And their lack of having an objective outcome measure of cognitive decline means that they are forced to use the actual levels of the target protein, okay, the drug being that's being targeted by the drug, as a surrogate endpoint. And the problem with this is that you are only assuming that this reflects actually the real clinical outcome, which is cognitive decline. And here is where synaptic markers could be a game changer and really affect the design of clinical trials and potentially even improve the yield in positive results. Because as I've shown from the data, synaptic biomarkers correlate with cognitive decline, as I've shown in Down syndrome and in dementia with Lewy bodies. What you measure in the CSF correlates well with cognitive performance. And in fact, going back to 1991, when it was first reported that synapse density is the best neuropathological correlate of cognitive decline. So it seems clear that we need to be able to measure the synapse degeneration in order to move forward and redesign our clinical trials. So this is something we are working towards, and we're in talks with pharmaceutical companies looking at whether we can measure our CSF markers in CSF that has come from the clinical trials to see if it really is a good measure of drug response. We are working towards transferring these biomarkers to plasma, as plasma would open up a whole new window of opportunity to measure this in a wide range of patients. And in particular, this would open up the exploration in psychiatric disorders where they typically don't do lumbar puncture and collect CSF, but they do have blood data available. And so we wrote a paper on the potential use of these synaptic markers in psychiatric disorders moving forward. And I think that would be a really promising avenue to pursue. So that's all. I hope that was not too much information in too short a time. Thank you for listening. And I would like to thank Alberto Yeo, who is the leader of our San Paulo memory unit, and of course, all of the members, both the clinical and the lab side, who have all contributed in some way to these data, and the patients who donate their time and samples that really enable our clinical studies to go ahead. So thank you very much. And if you have any questions.
Thank you very much, Olivia, for this very informative talk. I didn't know so much about all these biomarkers, and I was impressed of the potential that they have in the clinic and for the resolution or at least prevention of these diseases. I had a question, and that's if I remember correctly. I think that dementia is more prevalent in women compared to men, and I was wondering whether these biomarkers may differ between genders or may be more prevalent in one gender compared to another. Do you have any insight on this?
We have not so far seen any difference between male and female in these biomarkers. Unfortunately, the sample sizes are small, that when you get into so many subgroups, perhaps you don't have the statistical power to really to extricate these differences. But just based on dividing them male and female, we don't see any significant difference in the levels of these synaptic proteins. In terms of dementia being more prevalent in women than men, this is something that is often cited, and I think it's still being looked into, whether this is actually more a bias in the diagnoses or actually reflecting the actual biological basis or difference, like whether that's hormonal differences or genetic differences. But there's certainly a lot of data in the field, and I think a lot of people are looking into this. This is certainly a main objective now in many clinical studies, to look for sex differences. In fact, actually, there is sex differences in neurofilament light protein. So it's not a synaptic marker, but it is a marker of axonal degeneration. And in general, men, and there are many studies that are showing that men have higher levels of neurofilament light in their CSF than women. And it's unclear why, but it is believed that potentially it's lifestyle differences resulting in more neurodegeneration in male and female, but it's by no means proven or fully understood.
Mm-hmm. Really interesting. Does anyone from the audience have questions? Okay, so I had another one. So let me see, because I got one question. Yeah, so the dementia that is observed in Down syndrome, can it be compared to those patients that do not suffer from Down syndrome? I really don't know. I'm very ignorant. I'm sorry for this very basic question.
Okay, so the controls you mean, so they're non-trisomic, and so obviously there is a difference. They're non-trisomic, but what we were trying to show is that there is a difference. So Down syndrome adults who do not yet show AD changes, so the asymptomatic group, have low levels than you would find in age-matched people in the general population, so non-trisomic individuals. Now, this is possibly due to the Down syndrome itself rather than Alzheimer's disease, or it could be indicative that the synapse loss begins very early in Down syndrome and possibly even in childhood. Unfortunately, we cannot study this in children. This is an ethical limitation of the study, that we cannot look at these, do lumbar puncture in the children. But it would be very, you know, from a hypothetical or theoretical perspective, it would be fascinating to see whether this is something they were born with low levels of these synaptic biomarkers, particularly BAM2, or whether it's something that occurs very early in their development. As we see in the non-trisomic population, we see very early changes, perhaps in the second decade of life, which you can imagine in Down syndrome may be brought even further forward. But this is something we don't know. All we can say is that they don't correlate with intellectual disability, the degree of intellectual disability. So we don't think it is necessarily reflective of the Down syndrome itself or the intellectual disability associated with Down syndrome, but rather it is reflective of the AD process. And we don't know at what age that begins.
Mm-hmm. All right. Thanks a lot.
Can I ask a question?
Sure.
Very nice talk, Olivia. Beautiful data. I was interested, of course, on the FTD part, and where you showed that the calcitonin and urexin 2A were very potential biomarkers of FTD TDP. So, is there a way to look at the TDP-43 changes somehow also at the same time in the CSF?
Yeah, so CSF TDP-43 is something that's been studied a lot in ALS, and particularly in plasma. There are some new data coming out that shows that it could be useful in ALS in plasma. In CSF, it's not generally being used because mainly, so we have the problem in the clinic is that we can't tell the subjects that have the TDP. You know, so we can't define those that have it and those that have it not. It's true that now we have the synaptic biomarker that correlates with TDP as well. Maybe the combination of the two could be used to sort of neuropathologically subtype living patients, you know, so find clinical syndromes associated with TDP pathology. I think this is a really great avenue to pursue. And I think it's the way forward, because this heterogeneity in FTD between the clinical and the neuropathological data is so overwhelming that it's very difficult to really understand what you know, to define these patients in the clinic while they're living, which is why the cohort in Pennsylvania is so useful, because it has antemortem CSF and postmortem data. So I think you're right that looking at this in combination with TDP-43 could be very good, but when there's not strong evidence that TDP-43 in the CSF reflects what is in the brain.
That's true. Yeah. Thank you.
Okay. So I think that we can finish today's session. Thank you very much to our speakers for joining us today and sharing your research. It has been a very, very interesting seminar today. And also thank you very much to all the attendees that came here to listen to our speakers. Our next NeuroMeetup is going to be on so the first Wednesday of November, which is going to be the 3rd of November. And so then, until then, I hope that you have a good start of autumn, and see you next month. Thanks a lot again, Olivia.
Thank you very much.
Thank you very much.
Thank you.
Bye, everyone.
Bye.
Bye.